Hello, my name is Aissa Houdjedj, and it's such a pleasure for me to present our poster, which is titled Ranking Cancer Drivers via Betweenness-based Outlier Detection and Random Walks. Here we are proposing a computational approach, which is named BetweenNet, that integrates both genomic data and protein-protein interaction network to identify a set of driver genes that contributes to cancer development. And our approach is using BetweenNet centrality measurement on patient-specific networks to identify the set of dysregulated genes or a set of outlier genes for each patient. Then we also set up the relationship between the mutated genes and the outliers via a bipartite network. Then a random walk is applied on the graph, which provides our final prioritization of the mutated genes. Here, the main step of BetweenNet algorithm is shown in the following figure. For each sample, we will have gene expression measurement for both normal and tumor tissues, and also somatic mutation data from TCGA database. And in addition to gene expression and mutation data, we also employ protein-protein interaction data from INTACT database. The next step is the construction of personalized network. So for each sample, we will have two graphs, normal and tumor graph, that consist of the PPN network at the beginning. Then for normal graph, we remove non-expressed genes. Then for tumor graph, we also remove non-expressed genes and mutated genes as well. So in the construction of personalized network part in the figure, on the right part, we have the resultant graph after the removal of genes for patient P. Then we run betweenness calculations on both normal and tumor networks. So we will have betweenness values calculated from the tumor graph, and also betweenness values calculated from the normal graph for each patient. Then after betweenness calculation is done, across all instances for each gene I, we calculate the absolute difference of betweenness centrality values that are calculated from the normal and tumor network. After that, a bipartite graph is constructed that consists of mutations in one partition, in the top partition, and a set of outliers in the other partition. And an edge between mutated gene and an outlier is drawn if there exists such an edge in the PPN network. So to select a gene to be as an outlier in patient P, so what we do here is that for each gene I, we plot the betweenness values across all the patient. Then we estimate the standard deviation and the mean by fitting the truncated distribution. And a gene I is selected as an outlier in patient P if its betweenness difference values is greater than T standard deviation from the mean. After that, a random walk with restart probability is applied on the bipartite graph. So here the mutation frequencies of the genes are assigned as initial hit values to be diffused during random walk. Now, to rank mutated genes of the bipartite graph, we are using the following formula. We have alpha multiplied by the degree of the mutated gene I, plus 1 minus alpha multiplied by the weight of the incoming edges for gene I, which corresponds to summing the corresponding row of the random walk of the edge weights obtained after a random walk for gene I after convergence. And a gene I with largest score is selected as the next driver. Then we remove the corresponding node from the bipartite graph and its neighbors, and we recompute the scores again until we cover all the outlier genes. Then next, we compare our method based on their ability to recover the set of non-cancer genes. So as a reference set, we have CGC and CancerMine. So CancerMine is literature-minded database of cancer genes. We are including it here as it is a cancer type specific reference set. And CGC Rare contains the rare driver genes that corresponds to CGC genes with mutation frequencies less than or equal to 2%. So here we are computing true positive and false positive rates for the top 1000 ranked genes and calculate the area under the rock. and we are comparing our method against state-of-the-art cancer genes prioritization method. Here we have Betweenness driver net subsequency downrank and interdrivers methods on lung cancer, breast cancer, and pan cancer. And here we are showing the results based on lung cancer dataset. So as we can see that our method Betweenness is outperforming the other methods overall the evaluation. Then we evaluate our method based on functional and pathway analysis. So here we find the enriched category reactant pathways in the ranked gene sets of varying total sizes from 100 to 500 in an increment of 100 for each method, and we repeat the same category reactant pathway enrichment analysis with reference gene set. Then we calculate a consistency score, which is equal to the size of the intersection of pathways, enriched pathways, divided by the size of the union of pathways. Then for KEGG and reactome, our method is outperforming the other methods with a large margin for total size 100 and 300. And for GOAT term, we also observe that our method is ranked is outperforming the other methods for almost all total size values. Finally, as a conclusion, our main contribution is the identification of patient-specific dysregulated genes with a measure based on betweenness centrality, and also the employment of random walk have an added value in the identification of cancer driver genes. And we have shown in our publication that many of our top-ranked genes, which do not appear in reference cancer gene sets, have role in cancer development based on the existing literature. Thank you so much. This is my poster. Thank you.